Hi guys, so previous video we have learned what are the applications of DNA profiling. Now this video we're going to look what is the principle of DNA profiling. So before we go into check the principle of DNA profiling, let's check how the DNA is made up of. It's a double strand molecules, it's such a long molecules, and these molecules of DNA probably consist of many millions of base pairs. So base pairs mean here we have A pair with T and C pair with G. So likewise it's repeating many times in the DNA. If you take one cell, all the DNA, it could be a 6 billion nucleotides. Nucleotides are just like this one. That's a one nucleotide. So if it is have the base pairs, it could be a half of that one. It's a 3 billion base pairs. So these 3 billion base pairs make all our DNA in the cells. So they organize in the chromosomes. Now when you look at these DNA molecules, it's a quite huge molecules. Interestingly, 99% of these the DNA codes, they do not code for any proteins. So they don't make any proteins. Only 1% of our DNA make the proteins. So we call it a coding region, and this one is a non-coding region of DNA. So what is the use of this non-coding region of the DNA? So before we come to that, what is the use of this coding region of DNA? The use of coding region of DNA, they make proteins. The proteins is all our life. It make function and it make the structure. So it make everything the protein. So we will not talk about this one, but today we're going to check this part because it's interesting for DNA profiling. So 99% of our DNA are non-coding DNA. So what's the role of this non-coding DNA? The function of this non-coding DNA, so they have many functions, and interestingly most of the function we don't know yet. Only we know a little bit of few functions. So one function is simply we can say, when we learn the previous lesson, as our coding region of the DNA. Before coding region, we could have a region called a promoter. Onto promoter, the RNA polymerase bind, and maybe a little bit far away from promoter, either we have an enhancer or maybe silencer. Enhancer or silencer can again interact with the promoter region in cooperation with the transcription factors to regulate the gene expression. So here the promoter and these enhancer regions are out of this coding regions. So we can consider one function of the non-coding DNA. It serves as providing sort of a regulatory part of the DNA to regulate the coding region. Apart from that, the second major function is this coding non-coding region. So they can synthesize a RNA. For example, they provide the right sequence for tRNA, a ribosomal RNA, or maybe a regulatory RNA. For example, regulatory RNA, small interfering RNA. So small interfering RNA is the RNA which can really break down the mRNA. So for example, I just show here the mRNA. That's the coding region. So coding region produce the messenger RNA by process of transcription. So RNA polymerase is responsible to make this a messenger RNA. So as you know, messenger RNA can produce protein when it moves out of the nucleus into the ribosomes. So some siRNA break down the messenger RNA, so the RNA cannot make proteins. So we learned this one in epigenetics. That means how the gene expression is regulated, but the code are same, but the expression level they regulate the not to express the protein or to express the proteins. That's epigenetic one. That's another function of this non-coding region of the DNA. But the lesson is, what's the role of this non-coding region in terms of DNA profiling? So I think it's another third function maybe. It's not a function, but it's an application that we used to check the DNA differences, which we have used for the different applications. Now let's look at the non-coding regions. So these non-coding regions and the coding regions, that's here, that could be the coding region, and again the non-coding region, and again the coding region. So that's the non-coding, C for coding, that's non-coding, and that's coding. So likewise we have again I can draw just to make some ideas, not like alternative. So again maybe have a non-coding and a large non-coding, like it's a non-coding and non-coding. So we have all the non-coding regions here. So what we do now, this non-coding and coding regions, we are very standardized method of naming this, the introns and exons. So intron and exons, the introns are non-coding regions and exons are the coding region which transcript into messenger RNA. So we have a separate lesson previously. When they make mRNA, they make both introns and exons during transcription. However, during splicing by a spliceosome, they remove the introns and exon is only used for the translation process. So when you look at this non-coding region, so we can identify two important regions of the non-coding part. So what are the two important regions of the non-coding part? So just to show you, they are kind of a repetitive sequence. So what is a repetitive sequence? Let's say this one G, guanine, nucleotide, thymine and cytosine. This trinucleotide repeat like this GTC. So GTC, GTC, GTC. So that's a repetitive sequence. So this repetitive sequence, it's very unique in these non-coding regions. We can find large number of these repetitive sequence. Now, based on this repetitive sequence, we can identify two regions. The first region is called a mini satellite. Mini satellite. The other one is a microsatellite. So mini satellite and the microsatellites. So what are mini satellite and microsatellites? So we have another name for this one. Mini satellite is called variable number tandem repeats, and this one is a short tandem repeat. It's the same name, but just we have two different versions, like this way, mini satellite and microsatellite. Now mini satellites, they could have a 10 to 100 base pairs like this. Just I draw a sequence like this, minimum 10, GA, T, C, A, T, 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 G, C, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this one, this sequence repeat again like this G, A, T, C, likewise it's repeating. So that's 10 base pairs, and that could be repeat maybe 5 to 500 times. That's a mini satellite. So they have a quite large sequence which repeating again large number of times. So microsatellite, they have a short like just like a 2 to 6 base pairs. For example, T and G and T and G, T and G, like this. It's repeating many times, could be again 5 to 100 times repeating. So these are the regions we can see in non-coding regions. Mini satellite, they are 10 to 100 base pairs, repeating 5 to 500 times. Microsatellites, they are short sequence like 2 to 6 base pairs and repeating 5 to 100 times. That's the architecture of the non-coding regions. So these non-coding regions and very important for us in DNA profiling. In the next video, video number 3, you will see how we can use this the sequence of non-coding region in order to check the two similar or within the species two similar individuals.